Transcription. Transcription means the synthesis of messenger RNA with the help of an enzyme known as RNA polymerase, and this process takes place in the nucleus of the cell. That is, from DNA, the synthesis of messenger RNA takes place with the help of RNA polymerase. Three phases are involved in transcription, that is, initiation phase, elongation phase, and termination phase. First phase, which is known as the initiation phase. During the initiation phase, the binding of RNA polymerase takes place at the promoter region of the gene. Promoter region is present at the 5' end of regulatory region of a gene. This is the structure of gene in which regulatory regions are present at the 5' end as well as at the 3' end. At the 5' end, there is a promoter region present where the transcription starts, while at the 3' end, the terminator region is present where the transcription is terminated. That is the structure of a gene, and this section or this region is known as structural region, where alternative exon and intron are present. So, during the initiation phase, the RNA polymerase binds with the promoter region of a gene to start the transcription. Binding sites at the promoter region. There are different binding sites in the prokaryotes as well as the eukaryotes at the promoter region. Binding sites in the prokaryotes at the promoter region, there are two sites present in the prokaryotes. These are 6 nucleotide sequences. First one binding site is present at the -10 sequence, which is known as thymine, adenine, thymine, adenine, adenine, and thymine. This is the 6th nucleotide sequence. While the second binding site is present at the -35 sequence, that is thymine, thymine, guanine, adenine, cytosine, and adenine. This is also a 6 nucleotide sequence. While in the eukaryotes, also 2 sites are present. These are at -25 sequence, which is known as Tata box. That is the 4 nucleotide sequence. Thymine, adenine, thymine, and adenine. While the second binding site is present at -70 sequence, which is cytosine, adenine, adenine, and thymine. This is also known as card box. So these are the 2 binding sites present in the eukaryotes. RNA polymerase. Composition of RNA polymerase. There are 4 subunits that are present in the RNA polymerases. The RNA polymerase consists on 4 subunits that are alpha, beta, beta prime, and sigma. The first 3 that are alpha, beta, and beta prime, they are known as the core enzymes, and they help in the polymerase activity. While the fourth one, that is known as sigma, it provides the binding site, that is the binding, it help in the binding of RNA polymerase to the promoter region. During the initiation phase, the RNA polymerase binds with the promoter region through the sigma subunit of the RNA polymerase, while the other 3 that are known as the core enzyme, they help in the polymerization process. Types of RNA polymerases. There are different types of RNA polymerases in the prokaryotes as well as in the eukaryotes. The prokaryotes, which is a single cell, and this consists on only one type of RNA, while and the eukaryotes, three different types of RNA polymerases are present. That are RNA polymerase 1, RNA polymerase 2 and RNA polymerase 3. RNA polymerase 1 help in the synthesis of ribosomal RNA, while the RNA polymerase 2 help in the synthesis of messenger RNA, while the RNA polymerase 3 help in the synthesis of transfer RNA. So, during the initiation phase, the RNA polymerase comes and binds with the promoter region of the gene to unwind the double helix of the DNA to start the transcription. During the initiation, here this is the promoter region. Before the initiation site at the DNA, RNA polymerase comes and attach at the promoter region of the gene. After attachment of the RNA polymerase, the two strands become unwound. One strand acts as a template, that is antisense, while the other acts as a coding strand, which is known as the sense strand. And after unwinding of the two strands, a bubble-like structure is formed known as transcription bubble. So that is the initiation of transcription. Second phase, that is known as the elongation phase. During the elongation phase, as the RNA polymerase bind to the promoter region, the sigma factor is released because the sigma factor just help in the binding of RNA polymerase to the promoter region. After binding, the sigma factor is released and only the core enzymes that help in the polymerase activity. These core enzymes extends the polymerization of RNTPs, that are ribonucleosides triphosphate. The two strands of the gene, that is the DNA, and which one strand out of these two strands act as a template for the polymerization activity, that is known as antisense strand, while the other strand is known as the coding strand, which is called the sense strand. During elongation, the RNA polymerase keeps on moving from 5 to 3 direction toward the terminator region. When it becomes reached to the terminator region, here the transcription terminates. After the formation of the transcription bubble, the elongation phase started here. During the elongation phase, the RNA polymerase moves on the DNA strand from 5 to 3 prime direction, and as a result, the messenger RNA is formed with the help of RNTPs. So this is the messenger RNA that is transcribed at the promoter region, and this elongation process shall continue toward the terminator region. Last phase is the termination phase. During the termination phase, stop the synthesis of messenger RNA, and the terminator region consists on a series of guanine cytosine base pairs followed by a series of adenine thymine base pairs, and the part of messenger RNA that is transcribed in the terminator region. It forms a loop-like structure known as hairpin loop or guanine cytosine hairpin, followed by a small tail of adenine uracil nucleotide. The hairpin loop take part in the termination of the messenger RNA synthesis. After elongation, when the termination region comes, so here the GC hairpin loop is formed at the terminator region, and the messenger RNA is released from the DNA. That is, the DNA become wind to form the double helix, while this is the transcribed messenger RNA. 3 phases involved हैं: initiation, elongation and termination. During initiation, जो RNA पॉलीमरेज है वो प्रोमोटर रीजन के साथ बाइंड हो जाता है। प्रोमोटर रीजन हमारे पास यूकैरियोट्स में टाटा बॉक्स हैं, और टाटा बॉक्स जो है या प्रोमोटर रीजन जो है ये इनिशिएशन साइट से पहले जीन के ऊपर प्रेजेंट होता है। RNA पॉलीमरेज प्रोमोटर रीजन के ऊपर अटैच हो जाता है। आफ्टर अटैचमेंट सिग्मा फैक्टर रिलीज हो जाता है और कोर एंजाइम्स अल्फ़ा, बीटा और बीटा प्राइम जो है वो पॉलीमराइजेशन में हेल्प करते हैं। बाइंडिंग के बाद जब RNA पॉलीमरेज की बाइंडिंग हो जाती है तो डबल स्ट्रैंडेड DNA जो है वो अनवाइंड हो जाता है। एक हमारे पास सेंस स्ट्रैंड कहलाता है और दूसरा एंटी सेंस स्ट्रैंड कहलाता है। और जो अनवाइंडेड DNA रीजन है एट द प्रोमोटर रीजन, दैट इज़ ट्रांसक्रिप्शन बबल। नेक्स्ट एलोंगेशन फेज़, और एलोंगेशन फेज़ में हमारे पास जो RNA पॉलीमरेज है वो मूव करता है फ्रॉम 5 to 3 प्राइम डायरेक्शन। मैसेंजर RNA ट्रांसक्राइब बनाता जाता है उस टाइम तक जब तक कि टर्मिनेटर रीजन पहुंच जाए, और यहाँ पे राइबो न्यूक्लियोट्राइफॉस्फेट जो है वो यूज़ होती हैं फॉर द एलोंगेशन ऑफ द मैसेंजर RNA स्ट्रैंड। लास्ट टर्मिनेशन, और टर्मिनेशन में हमारे पास GC हेयरपिन लूप बन जाता है। यही GC हेयरपिन लूप जो है ये टर्मिनेट करता है ट्रांसक्रिप्शन को। एज अ रिज़ल्ट जब GC हेयरपिन बन जाता है तो ये मैसेंजर RNA को रिलीज़ कर लेता है, जिसके रिज़ल्ट में DNA फिर से वाइंड कर जाता है और मैसेंजर RNA ट्रांसक्रिप्ट हमारे पास बन जाता है।